Here is something most people never consider. The most powerful anti-aging chemistry your body can produce does not come from a pill, a powder, or a prescription. It comes from the contraction of your thigh muscle. And you can trigger it from a chair. If you have ever told yourself you are too old to exercise, too busy, or that your knees make movement impossible, what you are about to hear may change how you think about aging permanently. Because the science now tells us that your thighs contain one of the largest biochemical factories in the human body, one that produces hormones, protecting your brain, suppressing cancer cell growth, and resetting your cellular biology every single time it is activated. The first number you need to understand is this: starting in your 30s, the human body loses 3 to 5% of its muscle mass per decade. After age 50, that rate accelerates to 1 to 2% per year, with muscle strength declining approximately 1.5% annually. Between the 60s and 70s, by age 80, some individuals have lost up to 50% of the muscle they had in their prime. This condition is called sarcopenia, and it is not simply a cosmetic or fitness issue. Sarcopenia drives insulin resistance, weakens immunity, impairs brain function, increases fall risk, and raises overall mortality. It is one of the most dangerous and least discussed consequences of aging. But what most people do not know, what the research has only clearly established over the past 2 decades, is that muscle is not just a mechanical organ. Muscle is the body's largest endocrine gland. When muscle fibers contract, they do not only generate force, they secrete hundreds of biologically active proteins directly into the bloodstream. These proteins are called myokines, and they travel throughout the body to communicate with the brain, fat tissue, the liver, bones, the pancreas, and immune cells. The thighs concentrate 60 to 70% of the body's skeletal muscle mass. Stimulating the quadriceps, the massive muscle group at the front of the thigh, is like sending an activation signal to the body's largest myokine-producing organ. The downstream effects reach the brain. The most studied of these myokines is irisin. Irisin is produced when the protein FNDC5 in muscle is cleaved and released into the bloodstream during exercise. Its production is triggered by PGC1 alpha, a transcriptional regulator activated directly by muscle contraction. Irisin then does something extraordinary. It crosses the blood-brain barrier and enters the brain itself. Once inside, irisin stimulates the expression of BDNF, brain-derived neurotrophic factor, the most powerful known growth factor for brain cells. BDNF protects existing neurons, promotes the generation of new neurons in the hippocampus, and supports synaptic plasticity, which underlies memory and learning. Why does this matter so urgently? Because the hippocampus, the brain's primary memory center, shrinks at roughly 1.5% per year during normal aging and at up to 5% per year in pathological states like Alzheimer's disease. A 2025 study provided the first human evidence that irisin mediates the relationship between physical activity and hippocampal volume, meaning that thigh muscle stimulation directly contributes to preserving the size and function of the memory center in the brain. People with sarcopenia show consistently lower irisin levels, and the cognitive impairment risk among older adults with sarcopenia is approximately twice that of those without it. Beyond the brain, irisin also converts white fat into metabolically active brown fat, raising overall energy expenditure and protecting against obesity and diabetes. Research has shown irisin suppresses cancer cell proliferation, protects neurons from the mitochondrial damage linked to Parkinson's disease, and promotes bone formation to prevent osteoporosis. This single myokine operates across the brain, fat tissue, bones, and metabolic system simultaneously. Irisin is not alone. Cathepsin B rises in the blood after exercise, crosses the blood-brain barrier, and independently elevates BDNF while stimulating hippocampal neurogenesis. IGF-1 is released from muscle during resistance training and enhances hippocampal synaptic plasticity. 8 weeks of resistance exercise increases serum IGF-1 and improves hippocampal function in aging populations. All of these myokines work in concert, and all of them can be triggered by thigh stimulation, including gentle, low-impact seated movements. 3 practical routines and 3 supporting meal strategies. Follow that can be implemented starting today. The first routine is the seated leg raise basic set. Sit upright with your back against the chair. Slowly raise one leg until it is parallel to the floor. Hold for 3 seconds with your quadriceps actively contracted, then slowly lower. Perform 15 repetitions per leg for 3 sets. Total time is 5 to 7 minutes. This movement is safe for most people, including those with knee discomfort, as it is a low-impact, non-weight-bearing exercise. Adding light ankle weights or even a small water bottle strapped to the ankle increases the stimulus significantly. The second routine is the supported sit-to-stand sequence. Sit at the edge of a chair with arms crossed at the chest or extended forward. Rise slowly over 2 seconds, fully extend the knees and hips. Then lower back down over 2 seconds without fully sitting. Perform 10 repetitions for 3 sets. Research on frail older adults found that repeated sit-to-stand movements meaningfully increased knee extensor strength within weeks. Adding a brief pause when almost seated in isometric hold dramatically increases quadriceps activation during each repetition. The third routine is the wall sit or standing knee march. Stand with your back against a wall, then slide down until your knees are at a 90-degree angle and hold for 30 to 60 seconds. If this is uncomfortable, stand behind a chair for balance and alternate. Lifting each knee to waist height for 20 repetitions per side in 2 sets. Performing any of these 3 routines once daily, preferably 30 minutes to 1 hour. After a meal is the optimal window, since myokine levels peak immediately. Post-exercise and the effects persist for 3 to 24 hours. 3 meal strategies support and amplify this myokine response. The first is the irisin-boosting protein breakfast. Use 2 eggs, 100 grams of firm tofu, half a cup of cooked brown rice, a handful of spinach, and 1 teaspoon of sesame seeds. Eggs and tofu supply leucine, the amino acid that most directly stimulates muscle protein synthesis and forms the raw material for myokine production. Brown rice provides complex carbohydrates as exercise fuel, and spinach delivers magnesium, an essential mineral for muscle contraction that is depleted by exercise. Magnesium deficiency reduces both muscle function and myokine output. The second is the muscle recovery evening bowl. Use 1 portion of salmon, half a cup of cooked lentils, a handful of steamed broccoli, half a teaspoon of turmeric powder, and 1 tablespoon of olive oil. Salmon's omega-3 fatty acids reduce post-exercise muscle inflammation and enhance IGF-1 signaling. Lentils provide plant protein alongside polyphenols that reduce oxidative stress. Turmeric's curcumin suppresses chronic inflammation that blunts myokine receptor sensitivity. Olive oil's oleic acid supports cell membrane fluidity, facilitating more efficient myokine signal transmission. The third is the PGC1 alpha activation snack. Combine a handful of blueberries, 5 walnuts, and 2 squares of dark chocolate at 70% cacao or higher. The resveratrol and polyphenols in blueberries directly stimulate PGC1 alpha expression, the upstream switch that controls irisin production. Walnuts add omega-3 fatty acids and additional polyphenols. Dark chocolate's flavanols increase nitric oxide production, dilating blood vessels and improving myokine delivery to target tissues. Eat this 30 minutes before exercise or immediately after for maximum effect. Safety considerations must be addressed clearly. Anyone with severe knee arthritis or a history of joint replacement surgery should consult a physician or physiotherapist to confirm appropriate movement ranges before beginning. Keep the spine straight and avoid compensating by rounding the lower back during seated leg raises, as this creates lumbar injury risk. People with severe hypertension should monitor blood pressure carefully during isometric exercises, such as wall sits, as these can cause brief pressure spikes. People managing diabetes with insulin or blood glucose-lowering medications should monitor blood sugar before and after exercise, and carry a fast-acting carbohydrate source in case of hypoglycemia. The youth hormones your body produces are not waiting in a pharmacy or a supplement bottle. They are waiting in your thighs, quiet, dormant, ready to be activated the moment your muscle fiber contracts. A chair, 5 minutes, and one lifted leg. That is where cellular reset begins.